Good afternoon, my name is Zach Bird, and this is my presentation on transgenic fluorescence. In this presentation, I'll provide a detailed explanation of the experiment I performed, describe transgenesis in detail, explain the impact transgenesis has had on medicine, provide evidence of my understanding, describe the downfalls of transgenesis. What is a transgenic organism? According to the National Human Genome Research Institute, a transgenic organism is an organism that has been altered by the introduction of one or more foreign DNA sequences. These foreign DNA sequences come from different species. Transgenic organisms are produced in labs mostly for research purposes. How can we utilize transgenic organisms? According to an article by the NIH, we already make use of transgenic organisms with recombinant DNA. Some of the milk products we consume contain products produced by transgenic organisms. Transgenic organisms testing may also aid in the development of new medication and procedures. What will I be transforming? In this experiment, I will be transforming E. coli using ampicillin and piccolo plasmid. What is a plasmid? According to the National Human Genome Research Institute, a plasmid is defined as a small circular DNA molecule found within bacteria and other microscopic organisms. Plasmids are separate from chromosomal DNA and replicate them by themselves. Plasmids have a smaller number of genes compared to chromosomal DNA. What does piccolo stand for? According to Biorad.com, a pigloplasmid is a plasmid designed to replicate plasmid in bacteria. Select transformants use ampicillin resistance and regulate GFP expression. Piccolo is composed of the three following genes: ARAC, GFP, and BLA. What do those three genes do? According to BiologyInsights.com, the ARAC protein regulates transcription of genetic data. GFP is a protein that is responsible for expression of green light. BLA is an enzyme responsible for breaking down beta-lactam, antibiotics such as ampicillin. What organism did we extract the GFP from? Once again, utilizing the resources from Biorad.com, GFP is extracted from this jellyfish. It naturally produces GFP, which glows green under ultraviolet light. ARAC GFP expression. According to Biorad.com, the ARAC protein regulates GFP expression by detecting presence of arabinose. When arabinose is present, it binds to the ARAC protein and allows the RNA polymerase to transcribe the GFP gene. The three plates. According to the Bacterial Transformation PowerPoint, in this experiment I used 3-plate variants. Those variants are as follows: LB-AMP, LB-AMP-ARA, LB. How does ampicillin affect E. coli growth? According to an article by the NIH, ampicillin prevents E. coli bacteria from developing their cell walls. This results in death of the bacteria. Why did we use ampicillin in this experiment? Ampicillin acted as a barrier for entry in this lab. All the E. coli that were transformed were resistant to the ampicillin, whereas the untransformed were susceptible to the antibiotic. What was the purpose of this lab? This lab was performed to present the unbelievable adaptability of bacteria, the incredible art of transgenesis, and the ways in which we may impact our surroundings through science. The possibilities are truly endless. Procedures. Step 1. Label one micro test tube minus piccolo and another positive piccolo. Step 2. Transfer 250 UL of transformation solution into each tube. Step 3. Place tubes on ice. Step 4. Collect single colony of bacteria from your starter plate using a sterile loop. Insert loop into transformation solution and mix in until all residue is within the solution. Repeat for negative piccolo and positive piccolo. Step 5. Insert new sterile loop into plasmid DNA stock tube and withdraw a loop full. Mix the loop full into the positive piccolo tube. Close and return to ice. Step 6. Allow tubes to incubate in ice for 10 minutes. While tubes are incubating, label agar plates as follows: LB-AMP ARA plus piccolo, LB-AMP positive piccolo, LB-AMP negative piccolo, LB positive piccolo. Step 8. Heat shock bacteria. Transfer both positive piccolo and negative piccolo into water at 42 degrees Celsius for 50 seconds. After 50 seconds, remove tubes and place on ice. Incubate for 2 minutes. Step 9. Remove tubes and add 250 of LB nutrient broth and reclose. Incubate for 10 minutes at room temperature. Step 10. Mix slightly, then add 100 of transformation solution and control suspension into the appropriate plates. Step 11. Spread solution evenly on plate. Return lid and flip upside down. Step 12. Stack plates, tape together, label appropriately, insert into stack and incubator until next morning. Results. Based on the experiment performed within the video linked on Blackboard, I will list my observations from left to right. The LB minus Piglow plate had bacterial growth but no GFP reaction. The LB AMP negative Piglow plate had no growth and no GFP reaction. The LB AMP plus Piglow plate had growth but no GFP reaction. The LB AMP ARA plus Piglow plate had substantial growth and a GFP reaction. Discussion. Gene editing and its life-saving potential. Genome editing is a scientific technique that allows for precise modification of an organism's DNA. Researchers use genome editing to add, remove, or alter genetic coding to achieve a desired result. These results can include treatment for genetic diseases, cancer, and more. Techniques used in gene editing. According to articles by Nature.com and Britannica, there are 3 primary methods. Zinc finger nucleases. In ZFNs, modified proteins bind to specific DNA sequences and introduce double-strand DNA breaks to modify a sequence. Talons work in a similar fashion, but they are much more precise. CRISPR-Cas9. By introducing a guide RNA to direct the Cas9 enzyme, CRISPR-Cas9 modifies specific sequences in the most precise method yet. Applications in medicine. Monogenic disorders. Disease caused by mutations in a single gene, such as sickle cell, can be treated through CRISPR's revolutionary process. According to CRISPR, the FDA approved exa-cel treatment for these conditions. Vision restoration. According to an article by People, a young boy born with LCA recently experienced vision restoration through gene therapy. Cancer treatment. According to an article by the NIH, gene editing could be used to enhance immune cells and their adaptability to target cancer cells. CART cell therapy involves the modification of a patient's T cells to recognize and attack cancer cells and has shown promising results. Neurological issues. According to Reuters, recent research has shown that lipid nanoparticles can deliver mRNA across the blood-brain barrier. This is an incredible innovation and will pave the way for Alzheimer's treatment. Ethical considerations. Germline editing. According to Harvard, modifying embryos' genomes has inheritable effects that will impact future generations. As of this time, embryo genome editing is under scrutiny both legally and morally. Regulation. Regulation frameworks need to be established to effectively govern the safety and ethics of gene therapy. Without proper regulation, the possibilities are endless in positive and negative ways. Genetic pollution. According to the Times, genetic pollution contributes to the spread of genetic material that can contribute to disease. In this specific example, bathing sites in the UK are populated by materials that are capable of developing antibiotic resistance. This could result in massive loss of life. Citations.